All right, guys, we are just going to wait a couple more minutes before we get started so that more people can trickle in. All right, perfect. So we are going to get started now. My name is Nikhil, and just to let you know, we are doing this through MCAT 101. So if you guys do have any questions or you aren't part of the newsletter that we send out each week, just message me your email and I will get you added. But yeah, email either me or MCAT 101 if you guys have any questions. So the way we're going to do this is I'm going to go through the slides. If you have a question, feel free to just send it in the chat or unmute yourself when I'm at the end of a slide or section, and I will get to you as soon as possible. All right, so we are going to get started with genes because I want to do a basic overview. So what are genes? We know there are a lot of parts to our body, but there's several layers that we need to break it down into before we get to genes. Genes are basically sections of our DNA in each of our chromosomes that tell us exactly how to express a specific trait. And each gene is located at a specific location or locus on each chromosome. So what's a chromosome? A chromosome is basically the box or a package for the genes. And that's how genes are inherited. They're transmitted from parents to children as chromosomes. And you get one from each parent. So together they're called homologous chromosomes. So what's a genotype? A genotype is the actual code for each gene. And this code is a specific sequence of nucleotides. And the length or the length of the sequence can vary based on the gene, but the exact sequence of these nucleotides determines exactly how the protein that's made shows up in your body. But there's definitely different ways to express each gene, and those are called alleles. Alleles are the different sequences that code for the same gene. However, alleles have differential expression, so they show up differently in your body, whether it's visible or not. When a gene is homozygous, that means you have the same allele or same form of the gene on both the chromosomes you got from your dad and your mom. When they're heterozygous, it means that the gene has different alleles, one from your mom and a different one from your dad. However, there's also something called hemizygous, and this has to do with things like sex-linked genes. Because we get a different chromosome from our mom and a different one from our dad, specifically the X from the mom and the Y from the dad, and this is the only chromosome pair to be that way, it means there can be different genes on each chromosome. Since the Y chromosome is super tiny, the X chromosome is the one that includes most of the genes on the sex chromosome pair. So with a male, what you have is one copy or one allele for each of the genes that are on the X chromosome, because the male has a Y chromosome instead of a second X like a female does. So the allele that the male inherits on the X chromosome is generally the one that's expressed, and that's called hemizygous. Now we get to the actual expression of these genes. And for that, we need to consider the phenotype. And the phenotype is the physical manifestation of the gene, whether it's visible or not. It may just be an infinitesimally small part of your body, or it may be something that you can visibly see, like the color of someone's eyes or their hair. However, some of these genes actually have effects on more than one trait. Like some genes can affect both the color of your eye and the color of your hair, or they can affect the, I guess, the darkness of your iris, or the length of your hairs or the thickness of your hairs. And that would be pleiotropy, where multiple traits or phenotypes are affected by the same gene. So not every single gene shows up in everyone that has it, or is expressed, or shows a phenotype in everyone that has it. And that's measured by penetrance. Penetrance is the proportion of people that express a gene that also have it. So let's say in a population, we have 100 people, and only 50 people actually 100 people have that gene, but only 50 people actually express that gene or show a phenotype for it. That's not complete penetrance. That's, let's say, reduced penetrance, where not everyone that has the gene in their chromosomes exhibits the effects of that gene. There's also expressivity, which measures how strongly that gene is expressed. So, for example, let's take people with red hair. Now, obviously, you'll have seen people with varying shades of red, right? And that's an example of expressivity. Even if they have the same gene that codes for red hair, the amount to which each of them expresses it is different on an individual basis. So if we look at the image on the top left, in the top left section, everyone that has the allele, the mutant allele, shows it. That means it's completely penetrant because everyone that has it shows it. But since there's not a range of how strongly that black mutant color is, it's called narrow expressivity because everyone has the exact same level of expression of that gene. And we can compare that to things like the bottom left one, where we see complete penetrance, where everyone exhibits the black mutant gene. However, they do it to different levels of black or gray, because there's varying or broad expressivity. Any questions so far? I'll wait about 10 seconds or so. And feel free to unmute yourself during this time. All right, perfect. So the next slide is about dominance of these genes. Oh, looks like I might have a question. Perfect. Thanks, Brian. So there are several types of dominance, and dominance is pretty much exactly what it sounds like. It's the level to which an allele has control over another allele. Thanks, Brian. So when you have a dominant allele, it means that only, oh yes, the difference between gene and allele. Okay, so a gene is just a section of your DNA that is basically the code for a specific protein or a specific phenotype that you see. So it could be, like I mentioned before, it could be a specific protein like the keratin you have in your nails and your hair, or it could be the hair color. The allele is different ways to express that gene. So there are different types of that gene, right? So if we go with hair color, there's black hair, there's brown hair, there's red hair, there's blonde hair. Those are all different alleles for the gene of hair color. Basically, they're just alternate forms of that specific, of expressing that specific gene. Does that make sense? Perfect. So back to dominance. So dominance, like I said before, just means that one allele may have a certain power over another because you only need one copy of that allele. Remember, you get two copies because of two parents. So a dominant allele means that only one copy is needed for you to express that specific allele and not the other one that you get. And dominance is generally measured or generally seen in the letter that we give to a certain gene. Usually a capital subscript, like we see in the center picture with the IA, is given to dominant alleles, and a lowercase letter is given to the recessive allele or the lowercase letter. A recessive allele, on the other hand, means that you need both copies, meaning one from your mom and one from your dad, to express that particular gene or gene that way. So for this case, a lowercase I means that you don't have any antigens on the surface of your red blood cells. Now there's different types of dominance. Complete dominance just means that the dominant allele, when you have a copy of it, will always be expressed instead of the other allele that you can get from your parents. Incomplete dominance, on the other hand, like you see in the top left picture with the pink flower, means that both alleles are expressed and they're generally mixed together equally. So for this flower here, we have the red allele for the flower, for the petal colors, and the white allele for the petal colors. Since there's incomplete dominance, both are expressed and intermixed, and so the flower shows up pink. On the other hand, we have codominance. And codominance just means that both alleles are presented just like incomplete dominance. However, they show up in their own areas. Now, these areas aren't necessarily determined, but they will show up by themselves in a particular area. So you see in this flower, you have red patches or red patches and white patches. Does that make sense to everyone? I can wait another 10 seconds or so.
Could you please repeat the codominance part you just said?
Yes, no worries. So codominance is a lot like incomplete dominance in that both alleles are expressed. However, when they're expressed, they tend to do so in their own specific regions. Like you see in this flower at the bottom of the left image, you see sections of just white and you see sections of just red. It's not necessarily intermixed like the pink in the top image. Does that make more sense?
Yep, it does. Thank you.
Perfect. All right. So I just want to point out for the middle image, this would be, well, I guess we'll get to that in the questions, you'll see. So I do have a couple questions for you guys just to make sure that we really understand the concept of genes, alleles, and dominance. I'll give you about a minute for each question and just type the answer into the chat. And if you have any questions, please type it into the chat as well. Perfect. So I am seeing a lot of C's, and that's Correct. So hemizygous is only really relevant to when you have one copy of the gene. And so the copy of the gene you get is the one that'll be expressed. And that's different to genes on autosomal chromosomes, which are the 22 other pairs, where you get the same exact gene, maybe a different allele, but the same exact gene on the chromosomes you get from your mom and your dad. So you end up having two copies of that gene. All right, question 2. All right, perfect. So I am seeing these, and that is the right answer, because if everyone has the gene for red hair and they're expressing this hair in or the red color for the hair, that means it's fully penetrant because everyone that has the gene is expressing it. Now for expressivity, since we have largely varying shades or varying expression of the gene for red hair, it would be broad. All right, so in this case, the words variable and broad are pretty much the same. I just used it so it wouldn't really look monotonous in terms of narrow expressivity or broad expressivity in terms of the options. Does that make sense? Yeah, they're basically the same thing. You can use them interchangeably, and I just wanted to expose you guys to broad and variable as well. But yeah, here's the last question for that section. Perfect. So with this question, I'm getting a lot of B's, and that's the right answer. So the reason I put this question out there is just so that you guys get the difference between codominance and incomplete dominance. And since we see both of the antigens on the blood cell as not like a hybrid between the two antigens, but both as their own separate antigens, we know that it's codominant. Now, if we saw a like half square, half circle antigen, that would be incomplete dominance. Yeah, exactly. Like mixed AB antigens where it'd be like half square, half circle, something in between. And obviously, dominant would be either the A blood type or the B blood type over the O blood type. And recessive would just be the O blood type, which is why you have the little letters and the big letters for the O blood type and the A and B blood type, respectively. All right, perfect. So now we move on to the origin of genetics and genes and alleles. So we start with this man called Gregor Mendel. This man was a simple man. He was a friar and he grew peas. Yep, exactly, Brian. And so back to Gregor Mendel. He was like from the 1860s, and he basically grew peas. But one day he noticed that there were specific patterns among the pea plants that he grew, like a specific color of their flowers or specific seeds based on who the parent flowers or parent plants were and who the progeny plants were. And from there, we've gone all the way up to like pedigrees that track specific family related diseases or diseases in general that are passed down. And we've also gone to like whole genome sequencing where we follow, we can map the entire genomes and all the genes of different species, including humans, and things like DNA probes, where we can look for certain genes and tag them. But it all started with this man, and he came up with some very basic laws that were later proven by clearly experimentation or disproven in some cases. We'll get to that. So we can start with his first law, the law of segregation. And basically what he's saying is that genes, well, part of what he's saying is that genes have different alleles. And he saw that based on like the differing flower colors in the pea plants that he grew. And he also said, you get 2 alleles for each gene, 1 from each parent. And these 2 alleles from the parents segregate during meiosis. So the one you actually inherit from each parent is a single allele. And it could be either of the alleles that your parents have. He also said that there is a sense of dominance between these different alleles. So if the allele for white flowers were dominant and the pea plants, the female pea plant had a allele for white and an allele for purple, and it would express the white flowers when it went into its flowering season, just because the white flowers exhibited the white gene or white allele exhibited dominance over the purple allele. And so he saw that, and that was part of his first law. Now, the part of meiosis where this you can see all these statements is anaphase 1. And that's where you get the homologous chromosomes, ones that, let's say, ones that we inherited from our dad, which will give the red color, and ones that we inherited from our mom, which will give the blue color. And so they can pair up in any way, and they can be separated in any way. And that's what he meant by segregation. You get from one gene, you'll get one allele that could go either way. It's pretty much random. And when you pull these homologous chromosomes apart, that's when this entire process of segregation happens, where you separate each allele. And that's what you see here on the image on the right. You have 3 genes clearly colored and 3 pairs of chromosomes. And it's pretty much random which allele for the gene ends up in which gamete. And any of these gametes could be the one to fertilize the egg, or in this case, be the egg, and end up turning into the progeny. So that was his first law. Then we have his second law. And this second law really depends on a couple important concepts of our chromosomes. So our chromosomes have centromeres. It is at these centromeres that our chromosomes are held and pulled apart. Now, like I mentioned before, we have homologous chromosomes, one from your mom and one from your dad. We also have sister chromatids. Sister chromatids are, for this example, we'll use this blue chromosome in the image all the way on the left. Now, the left half of this blue chromosome has all the genetic material of that chromosome. The right half also has all the genetic material. So these halves are basically the exact same, and they're called sister chromatids. So when these chromosomes, sister chromatids, line up with their homologous pair, so the blue and the red chromosome, they form something called a tetrad. And that's when you have all 4 lined up like this. In that situation, sometimes the chromosomes get tangled. And since it's the exact same genes in the exact same locations on both chromosomes, even if they have different alleles, part of the chromosome can detach and attach to the other one. And that's what you see at the very bottom right. You see part of the red chromosome has attached to the blue chromosome and part of the blue one attached to the red one. And you can see with the letters that the alleles have been swapped as well. In this case, the alleles being the capital letters or the dominant versions of a gene, or the lowercase letters being the recessive versions of the gene. So you can see the recessive allele from the red chromosome has been swapped onto the blue chromosome, and the dominant allele from the blue chromosome has been swapped onto the red chromosome. So this whole tetrad and happens during the prophase 1 stage of meiosis, where chromosomes line up at the center of the cell for separation. And this whole process is called recombination or crossing over, because the chromosomes that you get from your mom and dad can swap genes. And what Mendel tried to get out of this is that these genes line up and they're inherited separately. So we have separate genes along this chromosome. So they're in very distinct locations and they're inherited independent of each other. So while you may get the dominant chromosome, like the allele with the dominant, the chromosome with the dominant alleles, my bad, the chromosome with the dominant alleles from your mother, since crossing over can happen and you can swap genes, you may also get a recessive allele from your father. So he's saying that all genes don't necessarily go together. You can trade an allele for a gene and inherit stuff from both your parents when you get fertilization. The one thing that we found out later about this rule is that there's this system called epistasis, where genes are actually linked. Some of them are transmitted together, which opposes what Mendel said. And we will get to that on the next slide. So what is epistasis? It's really that one gene masks or modifies the effect of the other. And so they're really just linked together in terms of inheriting them from your parents or expression. So we have an epistatic gene and a hypostatic gene. And to explain that, I will use the image at the top left. So we have two genes here, the gene for baldness and the gene for the color of your hair. The gene for baldness is the epistatic gene because you need it to be a certain way to express the hair color at all. Like if you have a gene for being bald, you won't even be able to express the color of your hair. So that's why, since the gene for baldness masks the effect of the color of your hair if you are bald, the gene for baldness is called the epistatic gene. And since the blonde hair or red hair, the gene for the color of your hair is controlled by the expression of the baldness gene, it's called the hypostatic gene. So there's several different types of epistasis, and that really depends on the allele of the epistatic gene. So when you have, in this case, in the gene for baldness, when you have a dominant allele that makes you bald, you mask the effect of the color of your hair. So this would be called dominant epistasis. On the other hand, if it were a recessive gene that made you bald, then it would be recessive epistasis, because when you had the genes for baldness from your mom and your dad, you wouldn't be able to express your hair color at all. And there are different ratios when we get to the dihybrid crosses that you will see show up in certain types of epistasis. So with dominant epistasis, you'll get a ratio of 12 to 3:1. So if the gene for baldness was dominant, then you would get 12 out of 16 times, or 75%, you would get a bald person. 3 out of 16 times, you would get, if let's say the gene for blonde hair is dominant to the gene for red hair, 3 out of 16 times, you'd get someone that expressed hair, but expressed blonde hair. And the final 1 out of 16 times, you would get someone that expressed red hair. And this is realistic in terms of today's society. You see a lot more hair than you see with a lot more people with blonde hair than you see with red hair. Not necessarily the baldness part though. We also have recessive epistasis, and that gives us a ratio of 9 to 3 to 4. And that basically tells you that you will have, and we can look at the actually we can look at the bottom left picture for that. So in the bottom left picture for Labradors, there are two genes that are linked, and this is the dark pigment gene and the color of the dark pigment gene. So the first one is if there's any dark pigment at all in the Labrador, and the second one is what color is the dark pigment in the Labrador. So regardless, if we get a recessive gene or recessive allele for the Labrador, then that means there is no dark pigment. So regardless of what color the dark pigment gene is in the Labrador, they will be yellow. And that's because they just don't have any dark pigment whatsoever. On the other hand, if we get a dominant phenotype or genotype for the dark pigment gene, that means there is some dark pigment in the Labrador. But the color of the Labrador is determined by the genotype of the dark pigment color gene. So if it's recessive, you get a chocolate Labrador, and if it's dominant, you get a black Labrador. So the ratio for recessive epistasis is 9 to 3 to 4. So since dominance just requires one copy of the allele to be expressed, it is most likely the one to happen. So you'll get out of 16 dogs, let's say come from a litter, you'd get 9 that were black, you'd get 4 that were yellow, and you'd get 3 that were brown, based on the dominance of each gene and the chance that each allele has to be transmitted. Now in epistasis, there's 2 types, and that's metabolic and signaling. Metabolic means, let's say something like breaking glucose down, right? That we see in glycolysis. And signaling just being like, let's say a nerve, like a signal transduction in your nerve. What we see in metabolic is everything is like the second to last bullet on the slide here, where each gene or protein activates the next one in the pathway. And that means the upstream gene is, or gene A in this case, is always hypostatic to the downstream gene, or gene B. And you'll see that in the Labrador image over here, because it is a metabolic process where you create the specific pigment for the Labrador. The dark pigment gene comes first before you get to the color for the, before you get to the gene for the color of the dark pigment. And that's, oh, sorry, my bad. That is the metabolic pathway. On the other hand, I don't have an image for this, but you have a signaling pathway. And what makes this different is signaling is usually self-regulatory. It's like homeostatic. You have an inhibitory gene where the expression of a specific protein or gene blocks the expression of the next one in the pathway. And what that means is the downstream gene is generally epistatic to the upstream gene. And so if you have, let's say, a mutant in gene B, you wouldn't even notice the effects of gene A in the phenotype. You just see that gene B was the only thing that was a mutant or messed up. Finally, for epistasis, you have 2 types. There's synergistic and antagonistic epistasis. And that basically means when you have 2 genes that are linked, like in epistasis, if the positive effect that they provide by themselves is not as great as the positive effect they provide when they're together, it's called synergistic epistasis, because you didn't expect that it's directly additive. Sorry, you didn't expect that the effect would be greater than directly additive. So let's say gene A makes you 10% healthier. Let's say gene B makes you 10% healthier. If you have both, let's say it makes you 30% healthier. In this case, it's not a direct additive effect. You'd expect 20% healthier, but since together they're 30% healthier, it means they're synergistic, they work together. You also have antagonistic epistasis, which is the exact opposite. It means they work together to give you a stronger negative effect. Let's say gene A makes you 10% more likely to get, let's say, cardiovascular disease, and gene B does the same thing. Now, if those genes are expressed together and you notice that together those genes give you a 30% likelihood to increased likelihood to get a cardiovascular disease, that's antagonistic epistasis. All right, I know that was a long section, but I do have some more questions for you guys. Perfect. So I am seeing a lot of A's in the chat, and anaphase 1 is the right phase of meiosis where you can see the law of segregation take place. And that's because in this phase, homologous chromosomes are separated, and you will literally understand or see how the alleles for the same gene are now being segregated and can be inherited separately. All right, the next question. Perfect. I am seeing a lot of C's, and chiasma is the right answer. So when you have the tetrads, as you see in this image here, the points at which the chromosomes actually cross over and they, I guess, let's say switch their connections is called a chiasma. And the last question for this section. And as always, let me know if you guys have a question. Perfect. Upstream is the right answer. And an epistatic gene in a metabolic pathway is upstream. All right, gotcha. No worries, Brian. So it's just a concept of epistasis because in metabolic pathways, you generally have an entire sequence of activating genes. So when, let's say, we go to this slide, for the coat color of the Labrador, which is a metabolic process because you are building up the chemical or compound needed to create the pigment in this dog. Every gene, it activates the gene after it. So let's say you create, let's say you have a recessive copy for the dark pigment gene. You wouldn't see any effect of the actual color of the dark pigment, right? And so we'd say the first gene, the dark pigment gene, is epistatic to the second gene, the color of the dark pigment. Because all you would see is the effect of the first gene, which is that there is no dark pigment for the Labrador's coat color. And so the Labrador will appear yellow. And I do notice someone raised their hand. Was that an accident or did you have a question? Oh, no worries, perfect. Any other questions before we move on to how we get different alleles? I guess I can give it like 10 seconds or so. Perfect, sounds good. All right, so we have mutations, and this is really how we get alternative alleles for a gene. So what is a mutation? A mutation is just a change from the original DNA sequence that you inherit for a particular gene. And there's many different ways where you can get a mutation. And we're going to discuss just a couple of them today. So one of the things I want you guys to know about are transposons. And in eukaryotes, which we are, there are a lot of, our genome is super large compared to bacteria or prokaryotes. So in the genome of ours, a lot of the space is taken up by transposons. And transposons are really just parts of our genome that can literally pick themselves up, move to a different place in the chromosome or in our genome, and put themselves in. And that really messes with our genes, as I'm sure you can imagine, if it just plops itself down in the middle of a gene. And that's a huge way of how we get mutations in our genes. Because by a transposon putting itself in the middle of a gene, you could get basically a deleted gene where you don't express this gene at all because of where the transposon put itself, or you could get a modified protein that comes as a result of this transposon dropping itself in. We also have things called point mutations or substitution mutations. And so as you can see in the top image on the left, that's where a mutation happens to a specific base of our DNA in a gene. So we have a silent mutation where a nucleotide is substituted for a different nucleotide, but it makes no difference because of this thing called wobble or the degenerate code that we have. And what that is each amino acid can be coded for by several different 3-letter sequences in the DNA. And that's just because if a single mutation like this happens, it won't mess up the sequence of the proteins or amino acids too much. That's just in there so we save ourselves when something happens like a point mutation. We also have an missense mutation, and that's what happens when you have a mutation like we see in the substitution mutation here, where it changes the actual amino acid that our cells read it as. And so instead of glutamine, as you see in the normal code, you get lysine. And that's called missense, when you accidentally or your body accidentally swaps in the wrong amino acid because of a mutation that happened in your DNA. And the last point mutation we can get is a nonsense mutation. And that's when you swap in another nucleotide, and instead of it coding for another amino acid like lysine here, it codes for a stop codon. And that ends the protein abruptly, or truncation as it's called, where the protein doesn't have the correct effect as it would if it were expressed normally. And that's called nonsense mutation. So we also have frame shift mutation, and it's called frame shift because it shifts the reading frame of our ribosomes. So when our ribosomes go through the RNA that we make, it reads it in specific 3-letter words. And these 3-letter words are called codons, when you insert a base or you remove a base or two from our DNA sequence, it shifts the sequence of these 3-letter words, so you get an extra letter added or removed from each codon. And you can see that clearly here. All they did was remove a nucleotide in the case of the bottom, in the bottom section of the top image, or they added an extra nucleotide in the third section of the top image. And you can see how that's basically changed the entire sequence of the amino acids after. Like you went from glutamic acid in the normal one to glycine in the insertion and arginine after. And then the deletion, instead of tyrosine, glutamic acid, you went to threonine, lysine, and arginine. So you can see that has a massive effect on the protein that's the product. Now we also have larger mutations, chromosomal mutations. And this can happen when you get a duplication of a specific gene, or you get a deletion of a specific gene, an inversion, or a translocation. And we can go through each of these. So a duplication is just when you basically repeat the sequence of a particular gene. And that means it gets expressed twice as much, or its expression gets messed up because the duplication happened in areas like the stop, where the stop codon is supposed to be, or where the promoter is supposed to be. Same thing with the deletion. If you get rid of the gene entirely, well, you've lost your ability to express that gene, and that gene may become hemizygous because you only have it on one chromosome. When you have an inversion, you usually mess up the parts of the gene that are closest to each other. So you see where the gene A used to go into gene B, now goes into gene E. If there was something important in the middle, that part is completely lost. And there's two types of inversion, and that's called paracentric inversion and pericentric inversion. The two, they really differ based on, does the flip or inversion depend on the centromere? Like, is it rotated around the centromere, or is it rotated without including the centromere? Para means without the centromere, and peri means it's rotated around the centromere. Now, mutations don't necessarily have to be all bad. They can be good. And you can see that in sickle cell disease. In some areas of the world, malaria is a strong factor in survival. But sickle cells are generally bad for humans in general because it reduces a red blood cell's lifespan. However, in parts of the world with malaria, it's actually a good thing, because when sickle cells, because of their shape, have a reduced lifespan, it means that malaria can't take over the cell for long enough to replicate itself properly. So because the sickle cell's lifespan is shorter than the time it takes malaria to take over the cell and use it for its own good. So that's a case where mutation, while normally is bad, is actually advantageous. However, you also have deleterious mutations, or mutations that are bad, like Huntington's disease. Huntington's disease is basically a disease that is caused by the duplication of a specific sequence in the gene that causes the gene to have an extra long protein, which messes up the protein's function. There's also inborn errors of metabolism that you can get from mutation, like PKU, or I hope I'm saying it properly, phenylketonuria, where a specific amino acid can't really be metabolized by some people because the enzyme that people use to do it doesn't work properly for them because the mutation has messed that enzyme up. And in that case, it can cause a lot of bad things like seizures. That's just one example of a deleterious mutation. So how does this work in the population? And I'm going to be talking about a couple of different species here, or populations. So since when you have alleles and you have genes that can flow between species, you get something called leakage. And that's the flow of genes between populations. And this, as you can see in the top left image here, this flow can lead to several different things. And at the top, you can see how the flow of genes just causes these two populations to blend together and fuse into a hybrid population. Or when you have a flow of genes between two completely different species, you can create something, you can create a like intermediary hybrid, like a mule, when a horse and a donkey are the populations. You get a mule, which has an intermediary number of chromosomes, because remember, you get one from your mom and one from your dad. So when horses and and donkeys have 62 and 64 sets of chromosomes respectively, the mule only ends up getting 63. And since the mule has 63, it can't breed back with a horse or a donkey, which means it is inviable. It can't create its own species. And that is a problem with hybrids as well. We do see that with some of the plants that we grow. So when you have genes like that and alleles that can be passed on between species even, we also get this thing called genetic drift. And the way I learned genetic drift is that it's the change in the gene pool that we see based on the way we sample the gene pool, or just the chance that has happened in the population. Say there was a massive forest fire for a specific species of tree, and it really wiped out so many members of the population. After the forest fire, if we went out and we looked at the genes in these trees, we may see a completely different balance of genes and alleles than we did before the forest fire. And that's genetic drift. It's just the normal ratios of a gene in a population through time. It may just change randomly due to chance events like a forest fire, or it may appear different because of a event like us sampling a specific subset, like we see in bias during surveys. That's pretty much an analog of genetic drift. So we know that the variability of alleles for genes is what really allows us to mate properly and have mutations that can both be advantageous or deleterious. So there are things called bottlenecks, which happen when a chance event like a forest fire wipes out the variability of the alleles in a population. And this can also be caused by founder effects, say people moving, our ancestors moving from continent to continent. When a group or a population of humans or animals or trees go from one place to another, you have reduced variability because of the low number of people or animals or plants actually moving. And that reduced variability means that there can be something called inbreeding. Inbreeding means that this low diversity means that the same alleles are inherited and passed on. And if any of these alleles get a mutation, that allele is still passed on, and it starts slowly taking over more and more and more of the population. And you get something called inbreeding depression, which means because this allele, there's very few alleles in the population, any deleterious allele has a massive effect, and it causes reduced fitness or reduced viability of the population to continue surviving. To oppose this, there's something called outbreeding. And outbreeding is where you introduce a group of unrelated individuals or animals or plants into a population to increase the variation of genes and increase the fitness of the population. And what this does is it makes sure that no one allele takes over the population, so that if that allele gets a mutation, it won't wipe out or reduce the fitness of the entire population. That was also a massive slide to go through. So let me know if you have any questions about that. All right, perfect. So I do have another set of questions for you guys. This is the first one. All right. So the answer to this one is B, pericentric. And I can explain that, no worries. So paracentric inversion means that genes are swapped without including the centromere or this gray circle that we see in the chromosome. So if the genes B and C were switched, that would be a paracentric inversion because the genes that are switched don't include the centromere in the middle. In this case, we have a pericentric inversion, which means the genes that are switched do include the centromere in the middle. And you can see that because the B took a pretty much 180 around the chromosome, same with the C, and the D took a 180 to come to the top half, and the E took a 180 to the top half. If, let's say, the genes rotate around the chromosome, think of it like a fan. If they rotate around the centromere, then it's pericentric. If they rotate individually on one side of the centromere, then it's paracentric. Does that make more sense? Perfect. It's great to hear. All right. And feel free to ask me any further questions if you have them. Here's the next question in the meantime. Perfect. In this case, C is the right answer. And I just wanted to ask this question because these types of mutations, point mutations, are very important, at least for genetics. And definitely, I'm pretty sure they'll show up on the MCAT as well. Understanding a point mutation is super important. And this is the last question for this section. All right, so for this one, it is B, and I'll go through each of these concepts again because they have some pretty detailed explanations at times. So genetic drift just means the tendency of a population to drift towards a certain allele for a gene. And that can happen because of chance events or just the way we sample the population. So it's not necessarily creating a hybrid, it's just seeing the proportion of a gene at any given time increase or decrease because of a chance event. So B, genetic leakage is the one that can create a hybrid. And genetic leakage just means that there's a flow of genes between two separate species or populations that can cause them to create an intermediate phenotype or a hybrid. A founder effect just means you are reducing the number of individuals in the population or the variability within the population. And that's a type of bottleneck where you have reduced variability that can lead to things like inbreeding. And inbreeding just means you have certain alleles that take over the population, and if they are mutated, can lead to a massive loss in fitness for the whole population. Let me know if that doesn't make sense. And since it is 7:30, I am going to try to speed things up just so that I don't take up too much of your guys' time. So I will move on. Finally, we get to crosses, and this is really the meat of analysis for genetics, and that starts with things like Punnett squares. Punnett squares are basically a way to measure the genotypic and phenotypic ratios or chance in a certain cross or like mating. So you have two generations. You have the P generation, which is the parental, and you have the F generation or the filial or progeny or children, that generation. We have a monohybrid cross, which you can see at the top left, top right here, sorry. And that's just measuring the proportion of the genotype and the phenotype for one gene. So let's say the top is the male and the left side is the female. You put the parental genotypes, and since we're using the law of segregation, you can separate them into their own transmission methods, and same for the father. So if you, once you cross them, you bring the letter down or to the right, and you see, okay, is it a dominant or recessive phenotype that I'm seeing here using the capitalization of the letters? That's the monohybrid cross. And what you'll see is if you do heterozygotes or one dominant allele and one recessive allele, as you see for the female or the left side, and you have the same for the male on the top, you will see a ratio of 1 to 2 to 1 for the genotype. So one homozygous dominant, which will be two capital G's that we don't see here, two heterozygous dominant, which will be a capital and a small g like we do see here, and one recessive phenotype like we see in the two small g's here. Okay, so it's been a while since I've taken the MCAT, but I will try to give you the ratios for dihybrid crosses, and they really shouldn't expect you to know beyond that. Like maybe know the phenotype while calculating, but you don't have to necessarily see which exact phenotype corresponds to a box on the grid. I wouldn't say there are necessarily any shortcuts to dihybrid crosses, if I'm being honest. It is something that takes a while. But for the monohybrid cross, the phenotypic ratio is 3 to 1. Yeah, they do show up in practice a lot. I'm sorry, it's been a while, almost like 8 months since I've taken the MCAT. But in the monohybrid cross, you get a ratio of 3 to 1 for the phenotype. So even if you get homozygous dominant and heterozygous dominant, they combine together to still show the dominant phenotype, even if their genotype isn't all dominant alleles. And so that ratio is 3 dominant phenotypes to 1 recessive phenotype. Then you have dihybrid crosses, which use, as you can see in the bottom right picture here, the concept of independent assortment, which assumes that genes are unlinked and they can be transmitted, each allele in them can be transmitted individually. So you get a random combination of alleles. So in that cross, you will get a ratio of 9 dominant for both genes, 3 dominant for one gene, 3 dominant for the other gene, and 1 non-dominant whatsoever. And this bottom right picture is a dihybrid cross that includes epistasis. And this is just like the coat color one, but in mice. The real concept of this picture is just showing you that there are 16 boxes for a dihybrid cross because there are 4 combinations of alleles for both for 2 genes. Then we have a test cross, also known as a Brack cross, where you try to find out the genotype of one of the parents by crossing it with a homozygous recessive parent. If you get a 1-to-1 ratio, like you see here in the top right picture again, you know that the parent has to be heterozygous because you'll never get a 1-to-1 ratio of phenotypes when you cross with a recessive parent if the other one isn't heterozygous. If you get a completely dominant progeny generation or filial generation, then you know that the unknown parent has to be completely dominant because they don't have a recessive allele to pass on to make it a recessive progeny. Finally, we have sex-linked alleles. And what's really important here is that you understand that one of the parents, the male, will not have a X-linked gene to pass on. And that's where we get the concept of hemizygous. So these 2 progeny here on the right that are males will only have 1 copy of the X chromosome. So whatever allele they get on that copy is the one that they're going to express, whether it is dominant or it is recessive. Like you see in the bottom right square here, for gene N, the male will express the recessive version because he has a lowercase letter, even if he only has 1 copy. And that's because this gene is hemizygous since it's sex-linked. And when you have sex-linked genes, you want to look at the subscript. So if you're writing a Punnett cross, and you want to use subscripts for the dominance or recessiveness of the gene on top of the X. There's also something in sex-linked crosses called a carrier, and that is a female that exhibits 1 mutated gene and 1 normal gene, like you see in the bottom left square. And a carrier means they don't exhibit the phenotype because they have a dominant allele to cover it up, but they can pass on the mutant phenotype to a male or a female in the next generation. Finally, we have something called gene maps, and this is another method of analysis. This is just a measure of how linked 2 genes are, and that generally has to do with the distance of the genes on a chromosome. So in the image on the top left here, we have 3 genes on 1 chromosome. The closer together they are, the more linked they are. The farther apart they are, the less linked they are, because at chiasma, when genes cross over, if a gene is close to another gene that's being crossed over, it's probably going to switch to the other chromosome as well, just because of how close it is. And that's measured by something called the recombination frequency, which is represented by the Greek letter theta, or just the 2 letters RF. And all that means is if the recombination frequency is large, these genes are less linked because they are further apart. Recombination frequency just means how likely 2 genes are to recombine and have a random assortment of alleles in the gametes. So in measuring recombination frequencies, you can get percents like this, like you see here. And these percents are a measure map units or something called a centimorgan. And each map unit or centimorgan represents a 1% chance of recombination. And so the map units that you get between 2 genes can help tell you the order of the genes when you find the distance between each of them. So in this image, they were able to find the order of the genes A, B, C, and D based on the distances between each individual gene. And this is something that is, it's a concept that might show up on the MCAT, and it is relatively easy to master in terms of getting the order right. So you shouldn't have to spend too much time on memorizing this, but it is, if it shows up on the MCAT, it will be super important to know exactly how to find the distance and order. All right, we are getting to the last slides. So the last thing I want to mention for this section is another concept of analysis, the Hardy-Weinberg equilibrium. So there are several assumptions in the Hardy-Weinberg equilibrium. I apologize if you guys can hear my fans now because my battery is running low on my laptop. Gotcha. You're not sure how to deduce the order based on the numbers in the diagram. So let's go back to the diagram for a second. You will see these, when you analyze the RF of these genes, you will see the percentages of how often these genes recombine. So let's go with the top two numbers for now. Not necessarily trial and error. Let me try to give you a quick example, all right? So let's say we found that the distance between genes B and D is 40 centimorgans or map units. You can do that, but it'll take longer is my point. Let me finish. Hold on one second. Let's say the distance between genes B and D are 40 map units or centimorgans, so you get a recombination frequency of 40%. And you also find that the distance of A and D are 25%. So you know that gene A has to either be here, closer to gene B, or it has to be on the other side of gene D, closer to gene C, right? And so you can measure that by testing the bottom two numbers. You know that the distance from A to C is 30, and you know the distance from B to C is 45. So if A is 30 map units from C and 25 map units from D, it has to be on the left of D, because if you put A on the right side of D, it wouldn't be 30 map units away from C. It would probably be like 5 map units away from C. Does that make sense? Can you guys kind of see how you can use math to figure out where exactly these genes lie? All you need is a set of RF values for some of the genes, and you should be able to work out exactly where the genes are. And I can definitely go over it with you in, yeah, exactly. They're not going to ask you to figure out the order of the genes without giving you some of these RF values. That's literally impossible. It'd literally be random decisions at that point. RF and map units. Yeah, in RF and map units are the same thing. Map units and centimorgans are the same thing, because RF is just like how often two genes recombine, which is a measure of how far apart they are. And map units basically represent 1% recombination frequency. So they are, RF is like a measure and map units are the unit for that measure, if that makes more sense. Yeah, no worries. But yeah, we have Hardy-Weinberg equilibrium, which is another method of analysis. And this method of analysis assumes 5 things, which are really important to remember. So it assumes that the population is large, so there isn't going to be a massive shift of genotype frequency. Yes, I can do that after the slide. So the population is large enough that the genotype the genotype frequency won't shift massively from one generation to the next. There are no mutations in the population, which is super important, because if you suddenly add a new allele, it messes up the equation and the calculation entirely. It also assumes that there's random mating. So there's no real choice between mates and between phenotypes. And that leads into the next point. There's no natural selection. No choice or no allele is more fit than the other, because we wouldn't be able to calculate allele frequencies between the generations if there was selection for a particular allele, unless we measured how strong the selection was. So to keep it simple, they said there's random mating because there's no natural selection. The last assumption is that there's no migration. There's no flow of new alleles and new genes into or out of the population to mess up the proportion. So those were the 5 assumptions they made to find out the allele frequency in a population from 0 to 1. And the allele frequency helps us find out the genotype and phenotype frequency. And these are the equations for both of those, where P is generally the dominant allele and Q is the recessive allele. And as you can see, they do that in a 2-allele system. So since if you have a homozygous dominant genotype, that means you have 2 P alleles. And that's why P squared equals the frequency of a homozygous dominant phenotype, so on and so forth. 2PQ represents the proportion of a heterozygous dominant phenotype because you get a genotype of the dominant and the recessive. And you can also flip them because you don't necessarily, you have the commutative property, and you can have a recessive allele first and a dominant allele second. So on and so forth for the recessive allele, where 2 recessive alleles means Q squared is the proportion of the recessive phenotype. All right, so here is the second to last set of questions I have for you guys. Sorry for taking up so much of your time. If you would like me to go faster, just let me know. Perfect. I am seeing C's, and yes, there are 16 offspring squares you'd have in a dihybrid cross because there are 4 ways that you can arrange the alleles for the parents. So 4 alleles or 4 different gametes for one parent times 4 different gametes for the other parent gives you 16 different combinations of gametes. And before I move on, I do want to repeat the definition of a map unit. A map unit represents 1% recombination frequency. So out of 100 times, only 1 time these genes happen to be recombined when they were passed on. That means these genes are super close together. So it's basically a unit for the measure that is map frequency, or sorry, not map frequency, recombination frequency. It's like we measure distance in meters, we're measuring recombination frequency in map units. Exactly, if something is 45 map units away, there is a 45% chance of recombination. Yep, no worries. And so here is the next question for you guys. All right, so the answer to this one is actually B. And that is because the farther away 2 genes are, the less likely they are to be transmitted together because there's a large chance that recombination can happen in the middle of them that would split these genes up. Does that make a little bit more sense in understanding why if the recombination frequency is large, then these genes are less likely to be passed on together? Yeah. So like, because recombination measures how likely these genes are to be separated or recombined during meiosis. So if a recombination happens more often, it means these genes are separated and combined in a different order more frequently. So there's less chance for these genes to be linked. So the next question is a bit of a hard one. It's more application, but just make sure to really look at the question when you try it. No worries, no worries. I might have phrased it in the not, and I guess I didn't phrase it in the most clear way. That's what I meant to say. All right, here's the next question. Think a little bit on this one. You might need a calculator, just depends, but make sure you read the question really carefully because it may catch you off guard. This is probably the closest that I will give you questions to like the MCAT level. All right, so the answer to this question is D, but I will work through this with you guys together to make sure that we all understand it. So the first step here is to understand that the frequency of the recessive allele is the number that represents Q. So since we know P plus Q equals 1, we know that P equals 0.6. However, we're trying to find the frequency of the dominant phenotype, which means we need both P squared and 2PQ because P squared represents the homozygous dominant genotype and 2PQ represents the heterozygous dominant genotype. Together, they represent the dominant phenotype. So when you do P squared, you get 0.36. Then you have to add it to 2PQ. And 0.6, or PQ is 0.6 times 0.4, which gives you 0.24. Now you need to double that, so you get 0.48. And then when you add it to the homozygous dominant proportion, which is 0.36, you should get 0.84. And I can go through that again, but let me know if you need me to or if you understand. It's a little bit of a trick question in that I ask you for the dominant phenotype, which is represented by the homozygous dominant genotype and the heterozygous dominant genotype. That's what's really important there. But yeah, I will give you guys another 15 seconds to, yes, exactly. The work is to calculate P squared plus 2PQ. And since you can easily find both P and I give you Q, it's a simple calculation from there, but the trick is to make sure that you understand that you have to include 2PQ in there. All right, so it sounds like everyone's kind of understanding that. So I will move on to the last couple of slides. Yeah, exactly. Thank you for saying that. So now I've got a couple slides left for you guys, and this is really based on the long-term process of, or point of genes and how they change between people. So we've gotten to selection, and selection is the process that makes, well, it's a way that shows how different alleles are not equal, regardless of if they're dominant or recessive. All alleles aren't necessarily equal in their benefit to survival. And we see that in selection. Darwin really noticed that when he went to the Galapagos and he saw birds with beaks of different sizes. He saw that some beaks were favorable in finding seeds and finding food, and some beaks were not. So he called it natural selection. There is a natural bias towards certain phenotypes or certain alleles to do better in the population. And if these mutations or variations turn out to be help the organism survive, they're called favorable. And if these variations are heritable, it means that you usually end up having more offspring, and that means that you are more fit. And fitness was basically a measure of the reproductive success and the amount or the frequency to which offspring reached an adult age. So if you had a lot of offspring and all your offspring reached an adult age, you'd be considering as having a lot of a high fitness. If you didn't survive very long, or you weren't able to have many children or reproduce many times, you'd be considered as having a low fitness. Now, there's several different ways on which natural selection can act to increase the fitness of a population. The first is stabilizing. And stabilizing happens when you select against an extreme value. And you can really see that in the birth weight of human babies. So when we really think about it, babies that are born super light or with a super low weight struggle to survive because they just don't have the nutrients, or they just don't have they aren't built, not built, but they don't have the availability of like fat that they can survive on for a longer while, and the lack of food. Or on the other hand, we have super heavy babies that take a super heavy toll on their mother, and it's hard for them to pass out of the birth canal. So there's a selection against that as well. So what you find is the majority of babies, regardless of species, but I'm just using humans for now, are within a certain range, and that's stabilizing selection. Next, we have directional resistance, and this is a focus on a selection towards one specific extreme, as you see in the top left image. A way that you can really easily see this happen is resistance. So let's say, we'll use bacteria for this example. Let's say in a population of bacteria, one bacteria evolves or gets mutated a resistance to an antibiotic we use. Now, let's say the other allele for that gene doesn't have a resistance to antibiotics. The selection is going to be immediate and strongly towards the bacteria with the resistance gene, and that's directional selection. Finally, we have disruptive selection, which is a little rare in that it favors the extremes and doesn't like the intermediate value. And that really happens when you have things like polymorphisms, where it's like little changes in the gene that can cause it to have can cause a population to have two different distinct phenotypes. And we will, you'll see that in one of the questions I come to later. Darwin saw this in the Galapagos with birds whose beaks were either super thick or super thin. And that really favored the large seeds in some seasons, or the smaller seeds that showed up in other seasons. And it really showed a selection against birds with a medium-sized beak, because they weren't able to open the large seeds as well, and they weren't able to open the small seeds as well as the small birds. So that's disruptive selection. And so after Darwin noticed this and other people noticed this as well, there are some theories that were made. So first we have the modern synthesis theory, or neo-Darwinism. And basically that combined Darwin's idea of favorable mutations or fitness, and they added mutation and recombination. And so basically what that shows is they added an idea that mutation can also be a negative factor. And any mutation may cause the individual to be more likely to pass on their mutation. They also had in the series something called inclusive fitness. And that adds more to the fitness we mentioned earlier, which was just based on reproductive success and how often the offspring made it all the way to adulthood. Inclusive fitness also counts things like the support to your offspring and to other members of the population. Because if you support your population and you support your offspring, they're more likely to survive and thrive. So that's something they notice, like societal factors like that. And that's the concept of inclusive fitness. Finally, we have a concept of evolution, which is gradualism or punctuated equilibrium, where selection for specific traits have happened at different times. And what you see here is in the bottom left pictures or image. Gradualism contends that the change or selection and evolution of a species or a gene happened super slowly, and this slow change built up to create a new species. And then after a long time, slowly creating another new species, and so on and so forth. Punctuated equilibrium, on the other hand, said that species were mostly stable, and in sudden changes or events, they suddenly evolved rapidly and created a new species. And that really is important when we consider the next slide here, which is speciation and evolution. So what is a species? A species is really just a group of individuals or a population that can breed together and form fertile offspring. Like fertile offspring, meaning these offsprings can then also reproduce. And these kinds of species form by reproductive isolation. And that can form by things like, there are two different ways, prezygotic and postzygotic barriers. And like you see in the top left image, prezygotic just means that these two groups or populations are isolated based on factors that prevent them from even forming a zygote. That's things like having a different breeding season or being physically isolated, like adaptive radiation, where a group of species or a single species can island hop going off of the mainland and you get a founder effect. And so these different populations on different islands slowly develop their unique mutations and frequencies of alleles, and so can develop into their own species. That's a prezygotic barrier. You can also have, for prezygotic barriers, reproductive parts that don't match up well anymore, and so you can't even form a zygote. On the other hand, there are postzygotic barriers, which prevent the zygote from turning into a mature adult member of the species. That's things like inviability. That just means that even if the zygote matures into an adult, it cannot reproduce like a mule. Or sorry, it can't. That's inviability means that it can't mature into an adult. Things like a recessive lethal gene, where a gene, while dominant, if you have two copies of the gene, it turns into a lethal form of the gene that kills an individual. And you might hear of that if you've taken genetics with the agouti gene in mice. Another postzygotic barrier is sterility. And sterility means, like a mule, you can't reproduce even if you are able to mature all the way to adulthood. So these are two ways that two populations of the same species can become isolated and eventually turn into different populations. Now, what people have seen is that there are several patterns of evolution. And evolution isn't the same as natural selection. Natural selection is just a method by which evolution works. Because by selecting for a specific allele, you are changing the makeup of a population, and so can cause them to become reproductively isolated. So in patterns of evolution, they've seen things like divergent evolution, where, like I mentioned before, two populations of the same species may be in slightly different areas, and these areas exert different pressures, selection pressures, like, oh, let's say an earthquake happens in one area, or one area suddenly becomes dry. And these different pressures cause these two populations to evolve in different ways where they become divergent and they turn into new species. There's parallel evolutions where two species in a completely unrelated environments develop the same way because the environments exert the same pressures. Like let's say there's a worldwide, let's say there's an earthquake or an eruption that's felt volcano eruption that's felt worldwide. That's an example of parallel evolution where the same effect is felt worldwide. And so the environments exert the same pressures that cause them to evolve along the same lines. And the last pattern is convergent evolution, where two unrelated species evolve in the same way because, again, their environments cause the same adaptations in them, like fish and dolphins being mammals. They evolve the same types of adaptations like fins to help them move through the water better. The last thing I want to mention, just before we get to the final question, sorry for taking so long, guys, is the molecular clock. And this is a way of measuring evolutionary time. Since we haven't been around forever, since we haven't you've been using the scientific method forever, we don't know how old species are or how long ago they diverged or converged. And so what we do is, to go back in time, we take the current genotype or genome of the species, and we find its rate of random mutation and how strongly their selection has occurred in that species. If we use those, we can work our way back after finding the number of mutations in that species. We can work our way back and calculate a certain time period where they diverge from another species. And that's evolutionary time being measured by something called the molecular clock. Now that's the last thing I wanted to cover. I have 3 more questions for you guys. I really appreciate you guys staying this long, but these are just for your own knowledge. So this is the thing I mentioned earlier where we would see a example of a specific type of selection. In this case, yes, this is disruptive selection because you can see that these moths are being selected against for an intermediate color like gray, and instead are being selected for the extremes like white and black. All right, here is the next question. Perfect. Like I mentioned earlier, natural selection and evolution are not equivalent processes. Natural selection is on an almost immediate time scale where the population increases the proportion of certain traits because it is an adaptive advantage at the time. Evolution is a much longer term process where we see natural selection happen over a period and traits take completely over the population or they completely disappear from the population and change it in a significant way. Now, the last question I have for you guys. So in this case, the answer is actually B, parallel evolution. And that is because you have two related plants or two related species in different environments that develop the same adaptations in response to the same environmental pressure. So those four qualities are what make it parallel evolution, because A, they're related, and B, the same environmental pressures in different environments cause them to have the same adaptations. If it were divergent evolution, you would see two related plants in different environments develop completely different adaptations. If it were convergent evolutions, it wouldn't be related plants at all. It would be two unrelated plants having the same adaptation. Does that make sense? Perfect. All right, so that is it. And I just want to remind you guys, we are a volunteering organization and a nonprofit. So please consider donating, because everything makes a difference for us, and please spread the word if you have a chance. Last but not least, please thank you, and please send us feedback or ask us any questions, or me. And if you want to be added to the newsletter, send me your email and I will get you added. Thank you guys for staying so long. I really appreciate it. I hope you learned a lot. And I will stick around for questions, but have a great day.